just switch my camera on to say hi and good luck. We've had a good series of webinars thus far, so
no pressure. Yeah. All right, well, we've hit the top of the hour. Welcome to this Target 2035, a probe for every protein kickoff webinar series. This is number 3 of 4. My name is Matt Hall. I'm a scientist at NCATS, the National Center for Advancing Translational Sciences at NIH, and I'll be moderating for you today. So in our first session, there was a real wonderful discussion about why this ambitious target has been set. And if you were able to join us yesterday, you would have heard a little bit about what needs to be done. And there were some wonderful examples of individual probe development, the science standards that need to be set around probe characterization, and things like target class profiling that are going to be essential as we think about the entire proteome rather than one target at a time. But today we're here to talk about some of the how can we possibly achieve this really ambitious goal of creating probes for more than 20,000 proteins in our human proteome, let alone all of the interactions and protein-protein interactions, for example, that might exist, positive and negative modulators of individual proteins. And so we're going to do that by talking about some of the technologies. How can we accelerate chemistry? How can we accelerate chemical biology to achieve this? And that's important because, for example, when we listened yesterday, all of the examples that we heard, or nearly all of them, were talking about proteins with function that can be measured because they have activity, screened using arrayed assay approaches. But when you've got 20,000 targets, those arrayed assay approaches really aren't going to cut it. They're not going to be scalable, at least by 2035. And therefore, and also, when you're relying on activity, those approaches aren't really useful for interrogating all proteins and finding those chemical start points for creating probes. And so we need to think about things like pooled libraries, affinity approaches, rather than relying on activity modulation, chemical biology, covalency, and you're going to hear about some of those things today. I suppose we could have had a session on the how of the biology. I don't think we've really nailed down how we can do the biology at scale. We could probably have a session, and we might touch on that a little bit today, on how to scale medicinal chemistry. If you've got start points for 20,000 proteins, how do you use MedChem to optimize all of those? So there's plenty of challenges in this field. And the good news is Target 2035 has a monthly webinar series that's going to go on till 2035, I assume, and we can talk about all of those problems. But probe development starts with discovery of chemical matter and the creation of platforms that can help systematize that to create probes. And we're going to talk about those new technologies today, both experimental and virtual. There'll be a 4th session coming up in a couple of days that's in a more friendly time zone for our Australian friends and everyone else in the Asia Pacific region. And you can find out more information on the website about that. For our session today, we have a Q&A box. If you're watching and have a question, put the question in there. After the first 3 speakers, we'll have a Q&A. In the second hour, we'll do exactly the same thing. And I'll pull out some of those questions to ask the speakers. Anything we don't get to ask, don't worry. Our speakers will put answers back into the Q&A box, so you'll see a written answer during the course of the sessions. And finally, we do have a Twitter handle for this meeting, #Target2035. Please use it and follow the conversations that are happening on Twitter about this scientific conversation. So as I said, we have an amazing set of speakers today, and you don't need to hear anything more from me. What I'll do is I'll stop sharing my screen here, and I'll introduce our first speaker. And our first speaker is Angela Koehler from Massachusetts Institute of Technology, and she'll be speaking about expanding the repertoire of druggable targets. Angela, hand over to you to share your screen and tell us about your science. Thank you.
All right, can everyone see my slides?
We can.
Great. So I'm very happy to participate today. I'm in the accelerating chemistry session, but I will talk more about chemical biology, I confess. Here's some disclosures. Some of the technologies I'll talk about today have been licensed into a company that I started, and then we also collaborate with a number of other companies to make use of the technology. So I'm here at MIT in a cancer center, and our research is really focused on trying to bring chemical biology to problems of relevance in cancer. And as we think about where to take inspiration for our favorite targets, we look in a lot of different directions, but one of the places where we look is the cancer dependency map, which has been developed by colleagues all over the world, but mainly colleagues across the street in this direction at the Broad Institute. Lots of data from colleagues in this direction from a number of pharmaceutical companies, including Novartis. And so this public resource enables folks to go in and look for potential therapeutic targets, look for predictors of patient response. This is a way of trying to use cell viability and perturbation using different types of molecules, drugs, other genetic perturbants, and to try and get a feel for different connections. And they make data available all of the time. So in our group, we may have chemists, chemistry postdocs, chemistry students who've been impacted by some kind of cancer. And in this case, I'm showing you an example query of the DET map for multiple myeloma. So when you do that, you'll get a list that comes up shown here. And you'll notice a number of genes will arise. And this one gene, IRF4, shows up several times from different data sets, CRISPR data sets, shRNA data sets. And in fact, if you look deep into this list, all but one of these targets are actually transcription factors, which are sort of the prototype of an undruggable target. So let's click on IRF4. And when you do that, your screen will be filled with information about enriched cell lineages, and you'll see that multiple myeloma comes to the top, which is reassuring given that's how we started our query. You'll get information about codependencies, copy number expression. But if you look over here, you'll see an image of this transcription factor IRF4. It's not considered to be a druggable structure, in part because it's very floppy, it's not an enzyme, and most of our colleagues will look at a structure like this and say, well, I can't do anything about it. So if you go further into the dependency map, it turns out that this large set here, the largest set of targets that are predicted dependencies in cancer encode nucleic acid binding proteins. Actually, the largest subset are RNA binding proteins, and then a smaller subset, maybe 46 proteins, encode classical DNA binding transcription factors, which have been sort of one of the hallmarks of a so-called undruggable target, in part because they lack enzymatic function, they lack traditional binding pockets, and they're often relatively disordered in key regions. And so as chemists, we just don't know how to design small molecules robustly for these types of targets. And so our lab is interested in trying to impact these more obstinate targets. So as mentioned already, you're hearing about a whole suite of technologies that are meant to try to demonstrate that a number of these emerging targets are actually indeed ligandable. Those ligands can be used as chemical tools or probes. They could start as, they could be starting points for actually developing therapeutics, of course. I'm going to focus on just one technology that's actually a relatively old technology that's focused on binding assays as an entry point into chemical probe discovery. So one thing I'll say, and Damien Young, who's part of this session, will laugh about that, you know, the pendulum swings back and forth all the time with respect to which types of technologies are in favor or in fashion for starting points for developing chemical probes. And while Damien and I were colleagues together at the Broad for many, many years, the last thing that you wanted to do at the Broad was to think about a binding assay as a starting point. It was all phenotypic, 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 but binding assays are cool again, in part because if you can develop a mechanistically unbiased binding assay, you can find things that bind to your favorite target, potentially a transcription factor, that have different functions. They could activate or inhibit your favorite target. And then, of course, binders can be converted into other types of proximity induction tools, such as a chemical degrader, to clear your target as well. So I think if there's a mantra in our lab, it's that we like to start with binding assays. And so one of the assays that we've used now for 20 years is an array-based assay where we print lots of different little compounds onto 3x1 glass microscope slides. And we were funded by the NCI through the Initiative for Chemical Genetics to develop this technology. We basically make these chips and supply them to collaborators who can run a far Western blot on a glass slide, where they come in with a solution of their favorite purified protein, incubate it with the chip, maybe containing 12 to 20,000 different compounds, and then they detect binding, often with an antibody, although we now have a whole host of other detection methods that will work. And so through this approach, you can sort of rapidly survey target tractability, say for a challenging target, and go off to the races. Thinking more broadly, the way we've used this in our lab is that we'll take whole panels of targets, histone deacetylases, 100 transcription factors, et cetera, and look for binding signatures. And then thinking about transcriptional targets, with my favorite targets, another thing that we've done with this approach over the last, say, 6 or 7 years is to focus on trying to run screens, target-directed screens, in a native environment. So if you have a transcription factor that's part of a larger complex, we then apply those cell lysates where your transcription factor is expressed, often with a tag, and we look for binders either to the target itself, shown here in blue, where the target is now correctly folded, it has the right post-translational modifications. So we think that that's just sort of a better snapshot of that typically floppy target when purified. But moreover, this has actually been advantageous because when we think about the target in its relevant complexes, we've been able to identify compounds that bind to nearest neighbors. And so we just published a paper in Cell Chemical Biology where we found a fairly selective CDK9 inhibitor in an androgen receptor focused screen. So over the years, we and others have used this approach to put about just over 40 different chemical probes into the literature using this strategy. So I'll just say a little bit about the chemistry. We capture the compounds using an isocyanate-mediated strategy. We basically make glass microscope slides with a terminal isocyanate, and we robotically print compounds to form about 100 micron diameter features on these glass slides. And we can, using this specific chemistry, we can capture a broad set of different types of compounds because there are a number of functional groups that are compatible with the surface capture method. And an in silico analysis of the MLP collection shows that about 75% of the library is isocyanate reactive. And then we've built a chemical library here at the Koch Institute collaboratively with other institutions to make these types of arrays. And so I'll just go through one quick vignette from our lab that focuses on a transcription factor, again, as a prototype of a challenging target. We've been interested in trying to target MYC for many, many years, and we've run many different types of screens to try to find robust chemical probes that are selective for MYC, and we haven't had a lot of success. So we could have zoomed out, looked at the larger network, and found that its heterodimer partner, MAX, seems to be an intriguing candidate for chemical probe discovery. MAX and MYC come together, form heterodimers that activate transcription, but MAX itself will engage a broader set of proteins to form repressive heterodimers and can actually bind to itself to form a repressive homodimer. So we use the technology I just told you about, screen purified MAX against a broad spectrum of different types of targets compounds. And out of a 45,000 compound screen, we identified 117 assay positives. We pushed these through MYC-driven transcriptional assays and identified a smaller subset that modulated transcription with potencies under 10 micromolar. This is a structure of one of the most intriguing hits. The little blue hydroxyl is actually the functional group that reacts with the isocyanate on the slide. We took that off and worked with another compound that had roughly similar potency and collaborated with colleagues at Stanford School of Medicine to evaluate how it impacted MYC-dependent cell lines. And shown here, you'll see 2 MYC-dependent cell lines relative to fibroblasts. We saw decent potency and moved forward with biochemical assays. We asked, does the compound disrupt Mc-Max interactions? And it did not. But we noticed that the compound appeared to be stabilizing the max-max homodimer in an EMSA type assay. So we ran a whole host of biophysical assays, including size exclusion chromatography, AUC, NMR, et cetera, and confirmed this notion that purified MAX was being stabilized in some way by this compound. And in fact, it also perturbs MYC-driven transcription. There are a whole host of assays that we use to confirm that in a broad spectrum of cell lines. And it turns out this compound is a mixed mechanism modulator. So by stabilizing this MAX homodimer, it can bind to the same EBOX sites as the activating Mc-Max heterodimer and competitively repress transcription that way. But by stabilizing this max-max homodimer, you shift the dynamic equilibrium of the system where you leave MYC in a state where for some reason it actually starts to go away. You alter the state of MYC by removing its friend away. And in fact, we do see that in a whole host of blot-based assays and global proteomics. And this appears to be proteasome dependent in rescue studies as well. And then finally, I'll just say that we've also used chromatin-based analyses, mostly ChIP-seq, in collaboration with Charles Lin, who was at Baylor College of Medicine. We also noticed that MYC will fall off of the genome by around 4 hours, whereas we can actually see MAX being stabilized, which is consistent with the mechanistic hypothesis for this compound. We've done much more with this compound just to try and clarify the relevance of this particular mechanism from a therapeutic standpoint, in vivo studies as well. And you can read the paper if this is of interest to you. But for us, this was really just a starting point to think about a wider set of transcription factors. We don't have time today to sort of tell you about all the things that we're doing now, but I just wanted to call out that we've formed a collaborative network with investigators at a number of institutions to think about using this specific technology to go after fusion oncoproteins that are pathognomonic in a broad spectrum of childhood cancers. And so by finding binders to these different fusion proteins, we're exploring a number of ways to interfere with those types of targets. And then finally, I'll just say, sort of coming back to novel types of targets, we've also engaged Arturo Vegas at Boston University to think about using this specific technology for a completely different type of challenging target, cytokines. And so cytokines, unlike transcription factors, are considered to be undruggable because they have very strong compact globular folds, and it's hard to find good binding pockets on these targets. And so they're typically modulated with antibodies and peptides and considered to be undruggable with small molecules. We identified a small molecule that bound to IL-4 and published this somewhat recently in ACS Chemical Biology. And now thinking more broadly about cytokines as a class, our labs are collaborating to look at roughly 40 pro and anti-inflammatory cytokines, generating binding signatures and taking advantage of that selectivity data to make choices about compounds that we would want to follow up on in downstream chemical biology experiments or potentially translational experiments. And so with that, our lab benefits from a sort of very diverse group: chemists, biologists, engineers, computational scientists, and physician scientists. And we've benefited greatly from a broad spectrum of collaborators and funders as well.
Thank you so much, Dr. Koehler. Wonderful. Okay, so we're going to move right along. And our second speaker today will be Dr. Jacob Bush from GSK. We'll give him a moment to share his slides. He's going to be speaking to us about reactive fragment platforms for the identification of chemical tools. Jacob, you there? Good morning or evening.
Hi there. Coming right in, hopefully. Just sharing screen.
Yep, we see your screen.
How's that?
Yep, and we see your slides.
Fantastic. Okay. So thanks very much. Yeah, so I'm Jake Bush. I'm a group leader in the chemical biology department at GSK, and I lead our reactive fragment platforms for the sort of rapid identification of chemical tool molecules for emerging targets of interest. So just to give a bit of an introduction, into why we're interested in these approaches. So it's worth highlighting that GSK has really doubled down on genetically validated targets. We've made big investments in human genetics and functional genomics approaches to help us identify genes that are strongly associated with modulation of a disease phenotype. Now, what these approaches are giving is lists of genetically validated targets, many of which are quite new and understudied. And we're then faced with the challenge of validating and prioritizing these targets for acceleration of those that are most likely to yield a medicine into drug discovery. So ideally at this point, obviously, we want to get hold of some tool molecules to build confidence that a small molecule interacting with that protein can have some disease modifying effect, at least in our model systems. And so here, as we've heard already, we're looking, like many others, to binding-based approaches such as DELS and ASMS, as these let us screen sort of millions or even billions of molecules very rapidly against these protein targets as soon as we've got some of that purified protein in hand. So our covalent fragment platforms are really complementary to those approaches in enabling us to start to probe the function of those proteins from a slightly different angle. So covalent fragments obviously leverage the ability of fragments to very efficiently cover chemical space to give us these sort of weak but highly efficient interactions that we can optimize into more potent probes. And we couple that with this covalent warhead to enable us to trap that fragment protein interaction, enabling sort of very robust and rapid detection of those interactions, particularly with these emerging targets where maybe we don't have any assays in place. That covalency can also give us a number of other benefits. We get increased activity through the fact that we're trapping what is actually quite a weak interaction and driving this equilibrium towards 100% occupancy of that pocket. And that then enables us to start to probe the function of liganding that pocket of the protein. And we've even shown that you can start actually to then use these quite crude tools in phenotypic profiling to understand how occupation of that pocket might affect your phenotype. And of course, if you couple that with proteomic profiling to understand all of the targets that these fragments are binding, that can really help us deconvolute the targets and the off targets and build confidence in that target phenotype association. So a lot of people are obviously getting interested in this type of work at the moment. There's a great review very recently from Asher Mullard in Nature Reviews Drug Discovery, where he highlights a lot of the quite exciting work that people are doing in pharmaceuticals and academia in biotech to leverage the use of these approaches. So today I'm going to give you an overview of two platforms that we've used. These are both working with purified protein, and then point a little bit to where we think the field is going in the future. So the first one is a cysteine reactive covalent fragment platform. So this case study we have here is using the E3 ligase HOIP, which has a cysteine in the active site. And we made use of this alpha-beta unsaturated ester here, which was published in this paper by Statsiuk. And this is designed such that the electronics of the fragment is decoupled from the reactivity of the warhead. And that gives us a really nice consistent reactivity throughout our library. And it turns out this is actually really important for us enabling the sort of confident and robust determination of hits from these screens. So it's quite a simple assay. You just have, we do this in 384 well plates, either one fragment per well or in pools. And we simply pipette the protein across the plate, incubate for 24 hours. And during that time, any fragments that can form an interaction with that pocket near the cysteine have the opportunity to get trapped, which can lead to a covalent modification of the protein, which we can then detect by a mass shift in that protein corresponding to the mass of that fragment. So this work was actually carried out by a postdoc, Henrik Johansson, who was in the Crick GSK Link Lab collaboration. And he screened a library of 200 fragments against HOIP and identified this compound 1 as enabling complete modification of HOIP in a time and dose dependent manner. He then wanted to explore the selectivity of this compound and ran it against a whole panel of other E3 ligases and dubs, and found that this compound 1 gives very remarkable selectivity, just labeling HOIP among this whole panel. And this contrasts quite strikingly with compound 2, which is a compound that people have used in the literature for studying HOIP, which actually is quite unspecific among this panel. So encouraged by this work, Henrik went ahead and explored the functional activity of this compound. So this is a polyubiquitination assay. where we're monitoring the formation of ubiquitin chains, which appear as ladders on this gel, and that are formed by HOIP. And you can see that in the presence of compound 3, which is this compound here, it's a slightly optimized form of 1, we get complete inhibition of that ubiquitination, demonstrating that this covalency is also driving a functional readout.
Jacob, sorry to interrupt. There's a bar across the bottom.
Oh, is that on yours as well? I'll hide it. Sorry, I thought that was just on mine. Thank you.
That's great. Thank you.
That's better. Yeah, thanks. Okay, and so Henrik then went on to profile this compound in cells by proteomics, and pleasingly was able to pull out endogenous HOIP from MCF7 cells. But what was quite striking here was that in addition to that, he pulled out just a handful of additional proteins. And so it really suggests that while this is quite a small and moderately reactive fragment, we're actually getting quite remarkable selectivity across the proteome with this fragment. So I think this study really nicely illustrates how these system reactive fragment libraries can quite quickly get us to these crude sort of tool molecules to start to probe protein function, and perhaps more importantly, provide starting points for the design then of more selective and more potent molecules to target proteins of interest. But limitation obviously is that we require a cysteine to be in the pocket of these sites. So we started thinking about how we might be able to expand the applicability of these approaches. And we were very inspired by a paper from Cravat and co-workers where they used photoreactive fragments in a cell setting to enable them to detect ligand-protein interactions in cells. And so we pivoted this slightly to a more sort of simplified and targeted approach, where here we're working with purified protein and screening then quite a large library of these fragments that are just linked to a simple diazarine photoreactive group. So this work was carried out by Emma Grant, who was a PhD student on the GSK Strathclyde collaboration program. And she synthesized over 500 of these fragments and then incubated these with the protein, once again working in 384 well plates with 1 fragment per well. And she found that if we irradiate these fragments, we can convert this diazarine to a carbene, which is a highly reactive species, and that can then insert into nearby bonds. So for all the fragments that are not bound to the protein, this is very quickly quenched by water, but anything that's bound to the protein has the opportunity to insert into a protein bond, which we can then detect by mass spec as a mass shift of the protein in the spectrum. So Emma kicked off by screening this library against BRD4, and it worked really nicely. She got no cross-linking from most of the library, some quite strong hits, giving really nice strong signal for cross-linking, and then quite a lot of hits that give this quite weak cross-linking yields. And this is actually quite crucial to this technology actually. So a lot of these turn out to be quite potent molecules, but that cross-linking efficiency can just be very weak due to quenching by water and so on. So at the moment we're exploring other chemistries to enable us to try to boost that cross-linking yield. But Emma went ahead and screened this library against a panel of 6 different proteins shown here as a heat map of cross-linking yields for each fragment. And what's cool is straight away you can see that we're getting selective interactions between particular fragments for the various different proteins. And this sort of builds confidence that we're really driving this by a recognition event. On top of that, we can see sort of known chemotypes coming out for particular proteins. We've got the dimethyl isoxazoles here for the bromodomains, aryl sulfonamides for the carbonic anhydrase, and even for KRAS, we were quite pleased for this sort of notoriously challenging to ligand target. We were able to identify a few binders which showed some chemical similarity to reported compounds in the literature. One of the real benefits of this Fabric platform and all these covalent fragment platforms is the follow-up studies that are available to us. So the first thing we can do is determine the site of cross-linking, which can be really valuable with some of these emerging targets where we don't have crystallography. So we simply digest the protein with trypsin, run the MSMS, and determine the residues that we've cross-linked to. And in this case, with KRAS, we find our fragments are binding to these 3 residues at this loop in the switch 1,2 region, which is right next to the binding site of those reported compounds in the literature that we were similar to. So this gives us some tool molecules with a known binding site, and we can then start using those to profile other tools that we might have got from other platforms such as ASMS, and we can run competition experiments to determine whether they outcompete each other, such as in this case, and therefore binding the same site, or don't outcompete and therefore must be binding to different pockets. And we can actually take that a step further, it turns out, and run these in a sort of at a range of concentrations to induce this dose-dependent reduction in cross-linking, which you can plot. This is quite clean IC50 curves. And Emma showed that actually with the bromodomains and a series of BET inhibitors, these Fragment-based displacement values correlate pretty well with the in-house FET assay that we run. So this Fragment platform quite quickly lets us sort of, with emerging targets of interest, lets us identify some tool molecules for particular pockets and start to use those to profile other ligands against those pockets. So that gives us two chemistries that we can use to start to probe these proteins. One being the cysteine reactive, where we get this benefit of the drive to 100% modification, which gives us that functional opportunity of a readout. But of course is limited to pockets containing a cysteine. And then to balance with that, we have the photoreactive approach, which now is applicable potentially to any protein pocket, but has this challenge of these low cross-linking yields that we detect. So based on this, I'm quite excited now about some of these emerging chemistries we're seeing, such as the sulfonyl fluorides, which have been shown to let us bind to lysines, tyrosines, histidines, and other residues. So then this really opens up the number of pockets that we can start to target with these reactive fragment technologies, while importantly maintaining the benefit of these electrophilic type approaches, where we can drive to that 100% modification and get some of those functional readouts. There's of course other chemistries coming out. We can think about oxaziridines for methionines and ACTs for the acid-based residues. And so what's kind of emerging here is a suite of chemistries that we can use to start to probe the various pockets on these proteins by various different mechanisms. So looking forwards, thinking about how we can start to use these approaches to tackle this challenge of expanding the liganded proteome. There's obviously two ways we can think about doing this, with purified proteins and in cells, right? So in terms of purified proteins, this is very much as we've discussed today, and really still relies on the protein expression and purification, which starts to become one of the key bottlenecks. But as soon as we have that in hand, we can deploy a number of different covalent fragment-based platforms, cis-reactive Fabbits or other, to start to detect hits that then enable us to perform follow-up studies to determine the site of binding, use these to profile other ligands. And then one area I haven't really talked about today, but I'm really excited about is high throughput chemistry. So we found that we're able to actually perform analog synthesis of our hits in plate-based format and then screen those directly against the protein without any purification, to enable us to rapidly explore the SAR around these hits and then drive towards more potent tool molecules. But looking forward, obviously, there's a great deal of excitement about what we can do with these in cells, right? So Ben Cravat and others have really pioneered this space, showing that you can take these reactive fragments, put them into cells, and they will form interactions with a selection of proteins across the proteome. We can then trap those interactions with the covalent warhead and pull out those ligands and identify all of the proteins that they interact with. So I think this brings into scope that sort of tantalizing opportunity of screening a whole library of covalent fragments against the whole proteome, to build this database of all the fragments in your library against all the proteins in the proteome, which then becomes a resource that we can turn to for new emerging targets and sort of pluck out tools to help us start to interrogate the biology further. Now, in terms of technology, I think the mass spec, we need to see a few advances in the throughput and the sensitivity of that technology, but I think it's definitely on the not too distant horizon and offers a really exciting opportunity for the dramatic expansion of the liganded proteome. So with that, I'll just thank everyone who's been a part of this work, particularly Emma, Henrik, David, and Johnny, who really drove these two platforms that I described today, and highlight the two papers in which they were published if you'd like to getting any more information on those. Thanks very much.
Thanks so much, Jacob. What fantastic. So we're going to move right along to the third of 3 speakers for this hour. And at the conclusion of this talk, we'll have a Q&A, including questions from the audience. Please throw any questions you've got into the Q&A box and we'll look at those and aggregate them. So my third speaker today is Damien Young from Baylor College of Medicine. And Damien's going to speak to us about systematic chemical diversity to enable probe and drug development. Thanks, Damien. I'll give you a moment to share your screen. And to unmute yourself. Damien, you with us today? I know you are, we already talked. Well, if Damien needs a minute to set up, Angela, can I ask you a question?
Yes.
Right. So there was a question in the Q&A box and you addressed a little bit of it. So can you say a little bit about how many compounds you put on a chip? And there's a follow-up about conformation rate, but I want to roll something into the first part of that question. What can and can't you put on the chip? I mean, are there limitations to the chemistry that's on that chip?
Yes. So I should say we've worked on about 30 different surface chemistries over the years. But we liked the isocyanate chemistry because it's promiscuous. And as I said, it enables us to capture a broader set of chemotypes. We usually aren't as interested in all carbon molecules that don't have reactive sites, but we use strategies to prioritize compounds from the wider pharmacopoeia that often have a broad array of potential isocyanate reactive compounds. So we want to actually have hydrogen bond donors and acceptors in multiple parts of a compound, and they often sit in different orientations. So we typically put anywhere from 15 to 20,000 compounds on a single chip, and in a normal pilot screen for us, we might go through 100,000 compounds. So that's a typical screen for us. Yeah, I mean, we can certainly, if we have a known binder where the only isocyanate reactive site is actually important for engaging the target that's known, we can absolutely break the interaction, right? All anchored assays have that liability, but because the isocyanate chemistry is promiscuous and because our compounds have multiple sites of reaction, they're typically sitting on the slides in different orientations. And so we think we can sample multiple binding modes.
Thank you. Well, we might follow up on that. I'm going to see if Damien's here. Damien.
I am here. And
Wonderful.
for whatever reason, can you hear me?
We can hear you.
Reason my current presentation is not. Okay, here we go. Here we go. I think we got it.
Wonderful. Yep, I can see that.
Angela, did you finish?
We're good.
All right.
Yeah, and we can. Yeah, right. We can follow up. So yeah, please go ahead, Damien. Thank you.
Okay, so thanks, Matt, for the introduction, and thanks for the invitation. I want to just say sort of what I've been doing for 4 years here is to try to develop a way of doing drug discovery at Baylor College of Medicine, which is a place that didn't really have a lot of drug discovery going on. So I firmly believe that most of drug discovery, most of the innovation happens in academia, but most of the aspects of having aspects to develop drugs to those targets that we find out in academia are not available in academia. So that's sort of what I wanted to develop. Now, if you think about how would you do this in a standard setup in a pharmaceutical company, high throughput screening, which is HTS, is pretty much the way that most drug discovery is done in academia. But that costs a lot of money. And if you want to develop that in an academic environment, that's going to take a lot of time. It's going to take a lot of resources. It's going to take a lot of money that pretty much academic scientists don't have to develop. So we have to think very differently about that. And so I've always thought about the fact that academic scientists have to think very differently in terms of how we innovate with drug discovery. How do we generate efficacious small molecules that will bind and modulate targets of interest that we develop or that we determine in academia so that we can move them forward for drug discovery. So here's what we've done at Baylor College of Medicine. We've developed two platforms. One is based on fragment-based drug discovery. One is based on DNA-encoded chemistry technology. We call it DECTech, which is really based on DNA-encoded libraries. So I'm going to talk about fragment-based drug discovery just because I think it actually encapsulates a lot of what we've heard in the previous session. So we can sort of generate the concepts for fragment-based drug discovery and carry it forward. And so, fragment-based drug discovery really relies on the aspect of there's a drug-sized small molecule, which is typically in the 500 to 600 molecular weight, that gets developed into a lead molecule. Fragment-based drug discovery works on a very different concept where you actually utilize a molecule that's about half the size of the drug-like small molecules. So whereas drug-like small molecules may be in the 300 to 500 molecular weight, fragment-sized molecules are about half the size of that. And it's been shown that through structure-based drug discovery, you can actually develop a very weak inhibitor. So instead of a micromolar inhibitor in the case of a high throughput screening hit, you can develop a millimolar inhibitor into a drug-like molecule. Now, there are a couple of concepts that fragment-based drug discovery has sort of shown over the last 20 years. One is that fragments actually are very high efficiency binders. So whereas on the left side, I'm showing you a compound that may not be binding to the active site at a very high affinity or high efficiency way. On the right side, fragments actually, because they're so small, they actually have to bind to their active sites in a very high affinity, or they have to utilize most of their scaffolds in order to bind to the active site. So the efficiency of binding is very high. And so that ultimately means that fragments bind with very high efficiency. And that means that they can be, if you keep the efficiency of binding, and that's the key, if you keep the efficiency of binding, they can actually be developed into optimal small molecules to bind to compounds. So the other concept that I like to talk about is that why think about fragments? Well, drug-like chemical space is very large. It's very large. And if we think of sampling that, it's a very daunting task. I've thought about this for many, many years. Fragment chemical space is actually much smaller than drug-like chemical space. It's exponentially smaller. So if you actually divide the number of atoms in half, you actually get an exponentially smaller number of computations that you can actually make with those small molecules. So you can fill that in, and you can actually sample that with a much better efficiency than you can do with drug-like chemical space. And so that's what sort of we've been thinking about. Now, when we got into this several years ago, or many years ago actually now, we actually thought about the fact that most of the molecules that people were looking at in terms of traditional fragments were highly monolithic in their structure. So if you look on the left, the traditional fragments encapsulated basically monolithic heteroaromatic flat fragments. Whereas on the right side, I will show you that compounds that actually nature makes. So if you look at all of the compounds on the right are FDA approved drugs actually. And these compounds are not flat. They're very three-dimensional in their structure. And so actually I have to say that Proteome B is not a FDA approved drug, but it's a cockroach hormone. But suffice it to say, all of the compounds on the right are three-dimensional in nature. And so the question that we raise is that, can we be thinking about generating novel three-dimensional fragments? Now, there's a path to fragment-based drug discovery, and it typically involves around taking a commercially involved fragment library. You get a hit based on a primary biophysical assay that we've heard in the previous studies. You typically would validate that, and then you want to get a structural study to show what it would take to have a chemist actually improve on that fragment study to generate a lead compound. So we thought that that was a limiting thing, right? So if you have to have structural based studies in order to show what it would take a chemist to do to better that compound, that takes a lot. So we wanted to think about a different way of doing this. So we described a diversity-oriented synthesis-based fragment library where you would actually get a hit, you would validate it, but you would actually get chemistry-enabled structure-activity relationships. And I'm going to talk to you about what that means in a second. And that would actually lead you to chemical optimization in spite of a structure-based study. So we were actually able to take a fragment that we got to a kinase called the GSK3 beta, and we made a lot of hits, or we made a lot of analogs to actually increase the activity, and we actually got to this compound. So we started with a very weak binding millimolar compound. We got to 18 micromolar. It took us about 3 weeks to get here, all right? So the actual thought that I had in terms of basically coming to Baylor is that what if we actually had all those compounds based on this initial hit in the beginning, such that if we screened those in 1 day, we actually would have gotten here in 1 day. And that's really what we wanted to do. So we wanted to be able to start with a compound and actually see SAR in a primary screen at the fragment level, which is something that has not been demonstrated in fragment-based drug discovery. So we came up with a concept called systematic chemical diversity, where we can generate in an upfront investment chemical synthesis that we can actually generate all the possible variation in molecular structure. Because fragments are small, we're able to actually generate all that SAR up front. We're able to actually show that we can see what makes or breaks, so a 0 or 1 in a binary way, what actually generates activity or breaks activity. So it gives you the ability to actually determine how you can see the SAR, and you're able to, right away, without an X-ray crystal structure, go into generating a new compound or a drug-like compound. So here's what we decided to do at Baylor. We decided that we would start with a well-known heterocyclic scaffold, piperazine. So it's the third most FDA relevant drug or scaffold that's seen in FDA approved drugs, has a lot of good properties that we thought that would be useful. But here's something that we thought that was interesting from a chemistry standpoint. There are not a lot of carbon substitutions on piperazine. So if you look at the piperazine drugs, there are not a lot of actual compounds that actually utilize the scaffold of the carbon. And so we thought that's an interesting way of moving forward with generating a fragment collection of piperazine compounds. So most of the piperazine is wasted. So we started with the piperazine and we said, well, let's put a handle in there so that we can do the chemistry to make the piperazine. Now, let's put a substituent in on the piperazine so that we can, diversify it. But there are different ways that we can do it regiochemistry wise. So we can make different regioisomers, but then there are stereoisomers that we can make. So for each regioisomer, we can actually make relative and absolute stereoisomers. And we can obviously do that for all the regioisomers. And then at the end of the day, we can do what people have always done at the end of the day, which is to diversify on the nitrogens. So we made a library that we are quite proud of, that we have applied to a number of targets now. But it involves 833 fragment molecules that involve regiochemistry, stereochemistry, and appendage diversity. And so this has been shown to bind to a wide variety of targets. And I'm sorry, I'm not able to talk about it. I didn't get the ability to actually talk about it for this talk, but we've applied it to a wide variety of targets at Baylor. And it's been shown to be, this library has been shown to be very effective. So I'm just going to give you one target that I can talk about, and that is related to a nuclear receptor. So we are interested with a colleague of mine of modulating neurogenesis in the brain. It used to be thought that you were born with all of the neurons in your brain when you were born, but actually it turns out that you develop about 700 new neurons a day, and they turn over and they're involved in learning and cognition. And it turns out that a target called TLX, which is a nuclear receptor, which is one of 48 nuclear receptors that is actually an orphan nuclear receptor, is involved in this process. So we wanted to determine whether we can actually modulate TLX with our fragment library to actually activate it to produce new neurons in the brain. So one of the ways that we actually have done that is we've done a biophysical screen that actually can be utilized to determine if we can agonize TLX. So what we basically want to do is design a small molecule that can bind the nuclear receptor and then generate neuroprogenitor cells. So it turns out that there is a known compound, and this is a paper that is under review right now. We've determined that there is a actually known, not FDA approved compound, but a compound that actually has been in clinical studies that we know that actually binds to TLX, that actually stimulates neurogenesis in the brain. And it's called NSI 189. It's under development by a company called Neurostim. And it actually agonizes TLX. So we were able to determine in this biophysical screen that we were able to take NSI 189 and show that TLX, upon binding to NSI 189, actually releases interaction with its co-repressor protein and takes up a co-activator protein. So we designed this screen in my lab. And so taking that fragment library in one day, and so this is what I want to take you back to. This is what enables chemists to actually be able to develop compounds in a very efficient manner. We were able to develop a screen, or based on this screen, we were able to actually screen this library and show that all this SAR, which basically involves the regiochemistry. So on the x-axis is 2,3, 2,5, 2,6. On the y-axis is the ability to interact with the co-activator protein. So if we can actually bind to the compound, or to bind to TLX and actually cause it to interact with SRC1, which is a co-activator protein, we can actually cause it to stimulate neurogenesis. So I'm going to show you in rapid fire, and please forgive me that we don't have enough time to go through this, but I'm going to show you just one snippet of what we were able to see and what is enabling for a chemist to see in one day of doing a screen like this. So 2.3, 2.5, 2.6, stereochemistry on the following scaffold. And on the bottom side, I will show you a substituent. On the top side, I'll show you another substituent, and then I'll reverse them in the other graph. So basically, I'm showing you 24 constitutional isomers. So in one day, we were able to go through phenyl acetyl, not much going on there. Benzyl acetyl, not much interesting there. Benzyl acetyl, phenyl, benzoyl phenyl. And then we get to this very interesting substituent where we add a phenyl on the bottom and a benzoyl on the top. And we show that the enantiomer is not active. Again, one day we were able to see this. So we were able to actually get through all of this chemistry that we put an upfront investment in right away and show that in a dose-dependent manner, we were able to show that we were able to activate TLX, which is going to be a compound that we're interested in moving forward with Alzheimer's disease and learning and cognition and major depressive disorder. So this was just a fragment screen. And I wanted to share this with you because I think that this is a very low level thing that we did, but we made the upfront investment that we were able to show that we were able to use chemistry to generate the SARs to show that chemistry actually matters in this concept of chemical diversity and molecular recognition. Last but not least, I want to say that we are able to apply this to DNA encoded library technology, and we have also an investment in DNA encoded chemistry. We are able to generate libraries and actually select them based on the same technology so that we can generate, we can actually select for the protein, and we can generate compounds that bind to that, and we can select based on the sorry.
So Damien, we want to make sure we ask.
My computer's freezing.
Yeah, we want to make sure we ask one or two questions for our panelists too.
Yeah. I'm almost done. I'm almost done. So we can sequence the compounds that actually bind to the compound of interest. And so we're doing that in a new way that we call fragments to Dell, where we actually can generate fragments. We can make DNA encoded libraries, and we can actually develop libraries based on the DNA encoded chemistry technology. So, and that's what I have to say.
Thank you very much, Damien. Wonderful. So we're going to move into a Q&A part of the meeting. Angela thankfully got to put an answer in earlier, and we may circle back briefly. Jacob, there are a couple of questions that are right now into the second part of our 2-hour webinar, which is really on accelerating chemical biology. You know, we can argue about what the definition of chemistry and chemical biology are. Maybe Alison can talk about that. Our first speaker is Alison Axeman, who is a member of the SGC Structural Genomics Consortium and at the University of North Carolina at Chapel Hill. Now, Alison's going to talk about design of the first selective chemical probe for the pleiotropic kinase CK2. Good morning, Alison.
Good morning. Thanks to the organizers for giving me the opportunity to tell you this story today. I'm kicking off the accelerating chemical biology session of today. And I'd really like to tell you about a collaborative project we've done with SGC Frankfurt, as well as the University of Western Ontario, on developing the first selective and cell active probe of the pleiotropic kinase, casein kinase 2. So I'll start with a little bit of background about CK2. CK2, there's been a plethora of publications around this kinase, but I would argue that its function still is not completely known. And that's because there's over 300 substrates of CK2 that have been annotated, and they're implicated in diverse pathways in the human body. And many of these pathways are aligned with cancer, cell growth and division, cell survival, those type of things. And CK2 is known to be overexpressed in cancer. And that's the reason why most of CK2 research has been centered in cancer biology. But as you'll see moving forward, the part of why CK2 biology is not fully understood is there's no good probe molecules to study this protein. And many of the studies that have been done have been using a suboptimal compound that lacks selectivity for this kinase, which has confounded biological results. So the most commonly used inhibitor for studying CK2 is CX4945, first described in 2011. There's been nearly 150 papers referenced that use this CX4945 as a CK2 inhibitor. In the 9 years it's been known or so, it has advanced to phase 2 clinical trials for multiple cancers. And that's largely due to the fact that it is an anti-proliferative compound and shows at least micromolar anti-proliferative activity in multiple cancer cell lines. It's potent in cells, and this is an example of an assay that we use regularly in our lab. It's a nanobrett target engagement assay. You heard about this yesterday was when Stefan talked. But basically this shows target engagement of both CK2 catalytic isoforms, alpha and alpha prime, by CX4945 and shows submicromolar affinity for CK2. Where really, we do have a potent molecule here, but really the problem comes in is that CX4945 is not what we consider a selective molecule. When tested against 403 wild type kinases at DiscoverX at 1 micromolar, we see that 28 kinases are inhibited more than 90%. So that really is confounding when you try to attribute phenotype to CK2 when so many kinases could be meddling. So we went to the literature to find a chemical starting point to begin our program, which started about 3 years ago. And we found this pyrazolopyrimidine scaffold that was exemplified by AstraZeneca. It binds to the canonical ATP binding site of CK2. Its selectivity is what we consider narrow. At 0.1 micromolar, it inhibits mostly CMGC subfamily kinases, so structurally homologous kinases, and potently at that. It has some anti-proliferative activity in micromolar, much like CX4945, and it inhibits CK2 downstream signaling, which is shown here with AKT being the substrate. It does inhibit its downstream signaling. So starting from this compound, which we call AZG, we designed a library of pyrazolopyrimidines, a small focus library, where we actually remade 6 exemplified pyrazolopyrimidines that AstraZeneca had shown in their patents and publications. They're all very potent on CK2. This is enzymatic data here. And then we diversified a little bit and made 6 compounds that were non-exemplified, that were either modest, just slight changes in the structure versus what had been shown, or more modest changes where we removed whole groups or and just made more broad changes. And so this is now orienting you back to AZG. This is our chemical starting point. It is in our nanobrett assay. It engages CK2 catalytic isoforms very potently, single-digit nanomolar. Now screening at a slightly higher concentration, which we're more familiar with at 1 micromolar, it inhibits 17 kinases greater than 90%. So through that library that I showed you, the medicinal chemistry optimization, we did identify this compound here, which we call SGC-CK2-1. It's a selective probe, and I say that because it maintains this affinity, a nanomolar affinity in cells for CK2, but now we're down to 3 kinases inhibited greater than 90%. And I'll go more into a little bit about these kinases in a bit. It does bind to the canonical ATP binding site. This was done at SGC Frankfurt, makes key interactions with the DWG motif as well as the hinge. This is an example of the curve for the nanobrett assay. And we made a negative control, which is a structurally similar compound that lacks CK2 affinity. We interrupted this valine 116 interaction by putting a methyl group here. Also caused elimination of the coplanarity that is responsible for this DWG motif to make efficacious interactions with the binding pocket. And what we see is CK2 activity is lost. Another interesting takeaway is this compound in our small panel showed no anti-proliferative activity. So we wanted to follow up on this, and I'll talk more about that. So CK2 is selective. Orienting you to CX4945, the compound that everybody uses in the literature, is the least selective compound of our library. 17 is our starting point, AZG. And what we see moving up, I've boxed here, our probe 24 and our negative control. They're amongst the most selective compounds in the library. This is now a deep dive into 24, where we see the 3 kinases inhibited by greater than 90%, or the two CK2s, and DRAC1. This is just slightly lower at 86%. So this data is here. This is a binding assay. So what we like to do is follow up in an orthogonal assay format. And so this is enzymatic data now. And what we see that is the CK2 data recapitulates in the enzymatic form. DRAC1 is found to be a false positive, so this is not a real inhibition. And then DYRK2 is our nearest, most potent off target. And what we see is actually 100-fold selectivity over DYRK2 in the enzymatic assay. And notably, moving to the DYRK2, now a cellular target engagement with DYRK2, this 100-fold selectivity is maintained in the cell-based system. So we have 100-fold selectivity over the nearest off target. So an important thing in kinase biology is to then say, okay, so it engages its target. What do we see? Is it actually cell active? Does it disrupt downstream signaling? And the way that we do that is typically through Western blotting. So this is in that HCT116 cell line, a colorectal cancer cell line, notable because we don't have any anti-proliferative activity in that cell line, but we do still see disruption of CK2 downstream signaling. So AKT here is the substrate. We're looking for dose-dependent reduction of AKT phosphorylation, which we see. Moving to a U2OS cell line, another CK2 substrate is EIF2S2. Once again, we see dose-dependent loss of phosphorylation due to treatment with our compound. Two notable takeaways is that we do see inhibition of downstream signaling, notably in a cell line where there is no anti-proliferative activity. And also that our probe is actually at least 10 times more potent in cells than CX4945. So 30 versus 10-fold here, or it's 30 versus 10 micromolar. So we're seeing a very large difference in potency. And thanks to Frankfurt and the Litchfield lab for these Western blots. So remind you again, I mentioned that our probe was not active in the HCT116 colorectal cancer cell line, which CX4945 and our starting point AZG were active, which is 17 here. What an interesting trend we see is if we look at the selectivity, so these are the most selective for CK2 in the blue box, none of these are showing any anti-proliferative activity. So it seems as though a selective CK2 compound is not anti-proliferative in colorectal cancer. So we wanted to really, before we made broad statements about that, we wanted to expand our profiling. And so we did that. And we did this externally. So we sent our compound, which is the probe compound in both cases, to a 140 cell line at prokinase, as well as the NCI60. And what we see is in the large cancer cell line, so I have an arbitrary line here. This is a dose dependence. So this is the IC50, a line of 1 micromolar, and this one was run at 10 micromolar. So here is 10% growth, 10% growth. And what we see here is that about less than 10% of the cell lines are actually responsive to our probe in the large cancer cell line. And once again, not very many are responsive in the NCI60 panel as well. Some of these are showing a little bit of lethality, but we did test the negative control in the same panel, and some of the same cancer cell lines were responsive. So it seems that it's something about the scaffold and not as much about CK2. One of the other takeaways is that blood and breast cancer seem to be the most sensitive to this compound if we're thinking more globally. So I think it's very timely that I wrap up with just one of the future directions for this molecule and this class of molecules. CK2 is an antiviral target, and I think everyone's aware of the Cell paper that came out earlier this year that really highlighted CK2 is important in SARS-CoV-2. And so, and also that Senwa Biosciences has been using CX4945 in this arena as well. And CK2 has been associated with EBV as well. So in collaboration with the Wilson Lab here at UNC as well, funded by the North Carolina Coronavirus Relief Fund, we are exploring this library with respect to coronavirus, and we have nanomolar activity in an MHV assay, and we are following up on those leads now, trying to develop this cell active tool into an in vivo active tool. So I wanted to keep it brief, but I'm happy to follow up with any questions that anybody might have. But I'd like to acknowledge some important people that helped with this project. David Drury and Kara Wells were really my partners in crime here at SGC UNC, helping with the chemistry and the biological results, generating assay data. I mentioned my colleagues at Frankfurt who helped with the crystallography and some of the Western blots, and the Litchfield lab has also helped with a lot of the CK2 biology, and there's ongoing experiments to learn more about how good this compound is versus all of the CK2 inhibitors that are out there. And then SGC is funded by a lot of pharma partners, and myself, I'm funded by a lot of different agencies, and I'd like to thank them as well. So that is all I have today, and I'm a little ahead of time.
Thank you very much, Alison, and that's greatly appreciated. Reminder to everyone, if you've got questions, please throw them into the Q&A box, and we'll have another Q&A session at the end of up to 2 more speakers where we can address those. So our next speaker is local to me, maybe not to you, Jordan Meyer from the National Cancer Institute. And we'll let Jordan share. He's already, that was very fast. And Jordan's going to talk to us about chemoproteomic platforms to validate and discover probe targets. Good morning, Jordan.
Hey, morning, Matt. Let me see. Is my audio is working?
Yep, we can hear you. We can see your slides.
All right. So I want to thank you all for having me and give me the chance to talk with this really special group. So Matt introduced me. My name is Jordan Meyer. I'm a senior investigator at National Cancer Institute's Chemical Biology Laboratory. And today I'm going to discuss the use of chemical proteomic platforms to discover and validate chemical probe targets. So here's an outline of what I'll try to cover in this fairly short 15-minute talk. First, I'll give an overview of chemoproteomic methods and the different flavors that can be used to study chemical probes. I'll then provide a brief vignette from my own lab, describing how we've developed and applied these types of methods to study acetyltransferase enzymes. And finally, I'll end with a summary and some ideas for how chemical proteomics could be potentially a useful part of the Target 2035 initiative, which hopefully will spark some discussion in the Q&A later. So I think we've heard a lot throughout this conference that really many drug and chemical probe discovery efforts flow from 2 dominant paradigms. These are really biochemical assays and phenotypic screening. So these 2 approaches, of course, have been incredibly successful, but they're also presented with a remarkable challenge by the complexity of a living cell. For biochemical screening hits, the question is, can molecules, even ones that are very potent and selective in a test tube, navigate this very different environment to find and engage their intended target selectively? For phenotypic screening hits, on the other hand, the question becomes, which of the billions of possible biomacromolecules that are present in a cell is a small molecule actually interacting with in order to drive that given phenotype? Now, both of these questions are really united by the fact that they require a way to reach into the unknown and start to study what small molecules are doing on a molecular level directly in living systems. So chemical proteomics, or chemoproteomics, is really a collection of strategies that have been invented to help answer these questions by enabling small molecule protein interactions to be studied directly within cells and proteomes. These chemoproteomic methods, of course, come in a variety of different shapes and sizes. In order to discuss the potential utility to Target 2035, I want to start off by giving a brief overview of the 3 main approaches and their complementary advantages. So the first chemical proteomic method I'll introduce is what's known as competitive chemoproteomic profiling. Here, promiscuous inhibitors that bind very broadly to an enzyme class of interest are functionalized with affinity handles, such as a biotin or an amine for resin-based immobilization. Upon incubation with a cell extract, these affinity reagents, of course, bind to all the enzymes which contain complementary active site machinery, allowing the enrichment, identification, and quantification of these different proteins by LC-MS/MS proteomics. Now, this active site-dependent enrichment mechanism is really important because what it allows you to do is perform competitive experiments in which a small molecule ligand of interest, labeled here with an L, is pre-incubated with a cell extract and tested for its ability to impede enzyme capture. In this way, the small molecule's competition profile provides a measure of its affinity and selectivity across many different, or potentially all different members of the enzyme superfamily. So these competitive chemical proteomic approaches have several advantages. First, they provide inhibitor selectivity data without requiring the synthesis of any type of analog of the inhibitor. So in theory, a single well-designed affinity capture reagent can actually be used to evaluate almost any different type of small molecule for its interaction with an enzyme family of interest. Furthermore, the nature of these experiments provide a very deep focus on what one would intuitively think are likely to be the most challenging proteins amongst which to gain selectivity. That is, enzyme family members that share highly homologous binding sites or active site chemistry. One of the main challenges of this approach is that this very bias can also, towards an enzyme family's subproteome, means that if you're using these types of approaches, you may miss off targets that lie outside of these enzymes class being profiled. Furthermore, these class-wide affinity capture reagents often can't be directly applied within living cells. The second chemical proteomic method that's been really useful for studying target engagement is what I'll refer to as clickable inhibitor profiling. Here, an inhibitor identified in a biochemical or phenotypic screen is derivatized with a latent affinity handle, such as an alkyne or an azide, and incubated with living cells. Upon binding to their protein targets, that small molecule interaction, of course, can be fixed either via irreversible reaction with the enzyme active site in the case of covalent inhibitors, or by introducing a second moiety that enables irreversible photocross-linking for non-covalent inhibitors, such as this gamma secretase photoaffinity probe shown here on the right. Following cell lysis, the click handle then allows these protein-bound small molecules to be ligated to biotin, enabling pull down and proteomic identification. Now, this clickable inhibitor profiling strategy really represents, I would say, the gold standard chemical proteomic method for measuring target occupancy, as it directly reports on the specific types of protein a chemotype of interest can interact with. Another major advantage of this type of approach is that due to the cell permeable nature of the latent affinity handles used, it can be applied directly to living cells and thus may better report on how a small molecule behaves in this native setting. The major challenges presented by clickable inhibitor profiling are really design. For many small molecules, it can be very difficult, sometimes impossible, to identify a suitable or synthetically accessible point where an enrichment handle can be introduced that doesn't completely destroy the small molecule's activity. Related to this issue, these strategies are relatively low throughput, as each clickable inhibitor analog actually requires individual synthesis, followed by biochemical or biological evaluation prior to being utilized. The third strategy I'll introduce are what are known as label-free methods. There are actually several technically distinct types of label-free chemical proteomic strategies that have been invented. However, the one that's been most overwhelmingly adopted by the largest number of members in the field has been the cellular thermal shift assay, also known as CETSA. In this approach, two cellular samples are prepared, one treated with drug and one not. In parallel, these samples are harvested, split into aliquots, and a gradient of different temperatures are applied to each. At high temperatures, proteins in these samples precipitate, leaving a lower fraction of protein within the supernatant. This can be quantified by Western blotting or LC-MS proteomics. Ligand binding can often serve to stabilize proteins against this thermal denaturation phenomenon, resulting in a thermal shift that itself then provides a measure of target occupancy. Now, one of the biggest advantages of CETSA and related label-free approaches is that there's no requirement for analog synthesis. So in theory, these types of methods can be applied to any native bioactive ligand, drug, or small molecule probe of interest. Furthermore, these approaches are potentially applicable to a very wide range of the proteome, giving them the potential to reveal unexpected off-targets. One challenge faced by label-free chemoproteomic methods is that not all ligand-protein interactions induce thermal stabilization. And those that do not, of course, are going to be invisible to these types of approaches. Furthermore, thermal shifts can be caused by both indirect as well as direct and indirect interactions with a small molecule. For example, changes in high stoichiometry PTM, such as phosphorylation, that are altered by downstream from inhibitor binding. This makes orthogonal validation of these types of thermal shift experiments very important. Now, in my last few minutes, I'm going to describe how my own lab has applied complementary chemical proteomic approaches to start to study small molecule acetyltransferase interactions. So acetylation is a reversible chemical modification derived from the central metabolite, acetyl-CoA. This modification is most well known in the context of histone lysine residues, where it plays a major role in the regulation of gene expression by modulating chromatin structure, as well as protein-protein interactions, and is catalyzed by lysine acetyltransferase, or KAT enzymes. However, in addition to histone lysines, enzyme catalyzed acetylation can actually influence the function of diverse biomolecules. These include RNA, where acetylation plays a role in ribosome biogenesis, protein N-termini, where acetylation helps regulate protein homeostasis, and metabolites, where acetylation is a critical step of many pathways, for example, in the biosynthesis of melatonin. Now, despite being an ancient and very well-known modification, I think one of the underappreciated challenges of acetyltransferase biology is that until very recently, there have been few high-quality small molecule chemical probes of these enzymes. So many of the first molecules discovered to act as biochemical inhibitors of acetyltransferase enzymes contain chemical liabilities, such as electrophiles and catechol, shown here on the left, which actually limit their utility as specific cellular probes to these enzymes. Just to emphasize what challenging terrain this area is, in 2017, a literature survey found that cellular target engagement, a really gold standard data point, of course, for chemical probe validation, at that point had not been reported for any small molecule lysine acetyltransferase inhibitor that was found in the literature. So in order to start to address this unmet need and open up new areas in acetylation biology, back in 2014, my group started to develop chemical proteomic approaches to study acetyltransferase enzymes. Our method uses analogs of the acetyl-CoA cofactor that are covalently bound to a lysine substrate surrogate. These bisubstrate inhibitors were first developed by the laboratory of Phil Cole, who's now at Harvard Med School, and they form very high affinity bidentate interactions with lysine acetyltransferase enzymes. Now, I'm not going to get into the detail of these probes or the applications we use them for due to time constraints. However, I did want to emphasize a couple of important design elements that might aid people watching this seminar develop their own chemical proteomic methods. So one is that these chemoproteomic scaffolds can really be tailored for specific applications. For example, clickable photoaffinity probes, such as those shown on the left, can be used for covalent labeling and gel-based profiling of acetyltransferase enzymes, while resin immobilization of these capture agents often enables more sensitive capture and enrichment for proteomic profiling. Second point I wanted to make is that these CoA-based chemical proteomic probes can report on the active site occupancy of many different types of proteins, including lysine, N-terminal, and RNA acetyltransferases, as well as a diverse array of CoA-utilizing metabolic enzymes. We attribute this broad capture potential to the high affinity that's afforded by bead-based polyvalent display of these ligands. We really think that these types of resin-based pull-down strategies can be really useful when attempting to construct class-wide enzyme profiling reagents, because they both increase the K-on and decrease the K-off of these capture reagents, enabling highly efficient enzyme capture even without covalent photo cross-linking. So just to demonstrate how these types of reagents can facilitate inhibitor characterization, we recently collaborated with Pfizer to use competitive chemical proteomics to study the selectivity of a small molecule inhibitor of NAV50, an N-terminal protein acetyltransferase that's involved in sister chromatin cohesion as well as mitosis. The molecule shown here on the top left of your screen is a substrate competitive inhibitor that was discovered by Pfizer through screening a DNA-encoded library against purified recombinant NAV50 enzyme. However, within cells, NAV50 exists as a member of a much larger multi-protein complex, and the ability of the inhibitor to engage NAV50 in this context, as well as to discriminate against homologous N-terminal acetyltransferase family members, was unknown. Using a competitive chemical proteomic approach, we incubated lysates with increasing amounts of the putative NAV50 inhibitor and evaluated blockade of capture by our CoA-based resins using Western blot. What we see using this approach is that capture of NAV50, but not other N-terminal acetyltransferase enzyme family members, is exclusively competed by this molecule in a dose-dependent manner, providing additional validation of its biochemical selectivity, but now within a complex proteomic context. We've also ran into cases where complementary approaches are really required to understand acetyltransferase inhibitor selectivity. For example, when evaluating remodeling, an acetyltransferase inhibitor that was reported in Science back in 2014, we surprisingly found that competitive chemical proteomic profiling didn't reveal any interaction of the molecule with the active site of NAT10, which was its reported target. Synthesis and gel-based profiling of a clickable remodeling analog really revealed to us the potential for the core scaffold of this molecule to covalently interact with many members of the proteome, prompting us to start to reevaluate remodeling cellular activity. So our group, as well as others, have determined that the modified RNA nucleoside N4-acetylcytidine, also known as AC4C, is actually the primary product of NAT10 activity. What we find is that when we evaluate AC4C levels using immunonorthern blotting, is that in contrast to NAT10 knockdown, which completely abrogates cellular RNA acetylation, remodeling doesn't have a measurable effect on AC4C levels in either ribosomal RNA or transfer RNA. So in this case, the combined use of competitive chemical proteomic profiling and clickable inhibitor analogs, followed by follow-up studies, were really all required in order to reveal the need for better small molecule inhibitors of this cell transferase enzyme. Okay, I'm running short on time, so I'll just wrap up with a very brief summary. Hopefully I've highlighted today how chemical proteomics can provide important insights into target engagement. Developing these approaches requires significant time and attention, and I think is a very important goal of chemical biology. Finally, I also hope that I've illustrated to you that complementary approaches can be really useful and are often required to get a full picture of a small molecule's target engagement profile. In terms of utility to target 2035, I think chemical proteomics has a lot of potential to play a number of important roles. First, in validating and invalidating the on-target activity and selectivity of probes. Another application of chemical proteomics that I didn't cover today, but that we heard about earlier in the session, is that these probes can also be really useful in generating new leads. For example, competitive chemical proteomic probes, such as CoA-based probe I introduced, can be turned into fluorescence polarization assays for biochemical screening, while clickable photo affinity fragments, such as those we heard about earlier, can be used to define new small molecule binding sites. And there's, of course, now potential for turning any high affinity binder, we think, into a selective degrader. Finally, in terms of what's needed, one challenge of this field is that relatively few chemical proteomic profiling reagents are commercially available, which I think limits their accessibility and utilization. Similarly, I'd say the field still lacks standards for open data sharing on the scale of things like the Gene Expression Omnibus and PDB for genomes and structural data, respectively. And the development of these types of resources could really extend the reach of these methods. Finally, I'd just thank my lab members who are involved in this work. Thank you for your attention, and I'll be happy to take questions at the end of the session.
Thanks so much, Jordan. Fantastic. So we have one more talk to go in this session before we get to Q&A. Reminder to put your questions into the Q&A box. Our third speaker is Dr. Michelle Arkin from UCSF, who is going to share her slides with us. Michelle will be speaking today about accelerating chemical biology using artificial intelligence. Thanks, Michelle.
Thank you. Yeah, thank you. So Matt, you can hear me, see my slides okay?
Yep, both.
Okay, super, thank you. Yeah, so today been a fantastic session. Thank you so much. And I'm looking forward to telling you a little bit about a different approach that we're learning how to take on how we can use artificial intelligence to rapidly accelerate chemical biology. So I know there's a lot of hype in AI, and so I want to talk to you also about how we're trying to handle the hype cycle. So we started off with the ATOM consortium, thinking about drug discovery and how current methods of drug discovery are long and costly and have a high failure rate and all these things that you already know. And so if you start with a target, and then this is pharma timeline, an academic timeline is much longer than this. You start with a lead and then optimize it towards in vivo properties and then safety properties and then get into human clinical trials. And if we're talking about precision medicine, where we want to start with a patient, this timeline and this failure rate of 88% is completely out of alignment with personalized medicine. So why is that? And how can we go faster and farther? So one of the problems that's identified is that we do these things sequentially. So first we look for activity, then we think about PK, then we think about safety, then we think about developability. And if we could just do these things instead of sequentially, in parallel, we could already speed things up. So if we think about multi-parameter optimization, and this is something that computers are really good at. So capturing all this in the ATOM pipeline, I'll talk about ATOM in a second, we think about these in terms of property prediction models. So ATOM is a public-private partnership. ATOM stands for Accelerating Therapeutics for Opportunities in Medicine. And it's a public-private partnership that aims to accelerate drug discovery using high-performance computing. So high-performance computing and artificial intelligence require a lot of data. So they're data-driven models mostly. We also use structure-driven models, but largely data-driven models. So we need diverse biological data. And the type and quality of that data is really important, as is obvious to experimentalists. And then emerging experimental capabilities. So how can we take high performance computing, which can identify molecules, say, really quickly, but those molecules don't have any true activity until they're tested. How can we catch up and have our emerging experimental capabilities be as fast as the ability to predict an activity? So the group of people that have been involved with this so far are 4 national laboratories, starting with Lawrence Livermore National Lab, Frederick National Lab, Oak Ridge, 5 national labs, Brookhaven and Arkin. Us at UCSF and other academic groups are getting involved, and GSK. And we have 3 additional companies that haven't yet been disclosed, but we've been working with. So the ATOM Consortium is built around this set of activities that moves computation to the forefront of drug discovery. And as an experimentalist, having me be kind of the thing of last resort is sort of hard to get used to, but we're starting with a working compound library. So how we get that working compound library is something I'll talk about in a second. But we start with some molecules that have some set of properties. We then say, well, what properties do we want them to have? And we run those compounds through a set of prediction models, this efficacy, safety, PK, and developability. Having these predictions, we then say, how do those predictions match up to a set of design criteria? Insofar as they don't match up to the design criteria, we then run them through a generative molecular design loop. So this is where you takes the molecules, it converts them into something that a computer can manipulate, make a bunch of new molecules that are predicted to have better properties, go back in, predict those properties, go back through the design criteria. And this goes through thousands of times. When the system either gets to a compound or set of compounds that are apparently have made the design criteria, met the design criteria, or the system says, I've moved into a chemical space where I don't have a lot of data, I can't make a good prediction, then we go into an experimental loop. Okay, we've met the design criteria, we want to test that, or we know that we can't predict the design criteria very well for a certain set of molecules. So we make them and test them. So can we make them quickly enough and test them quickly enough to be able to retrain those property prediction models and go back into the computational loop? So that's the technical goal. What's the conceptual goal of ATOM? So the goal of ATOM is to create an ecosystem for open drug discovery research and tools in artificial intelligence and then actual experimental biology. So starting from the foundations of advancing the science of AI in drug design through rigorous and open research, building a community of people that are involved in artificial intelligence and drug discovery, or are chemical probe or drug discovery people themselves that want to use these tools. We aim to build a community through open platforms and partnership-based R&D. Then the pillars, the activities that sit on this foundation are developing open curated data sets and open models in AI, simulation, and computing. Also basic research and performance and uncertainty. So that's that active learning loop that I was saying. If you know you've moved into a new space, how do you predict your inability to predict? And then automated experiments. So on top of all of these activities, the goals are pre-competitive active learning molecular design platforms, and a number of these platforms have already been put into GitHub, into the public domain, and open drug discovery for public good. And so I want to add here rapid chemical biology for public good. So this workflow is based on machine learning largely. So this is the same workflow. I'll keep coming back to this. There are 2 different ways of thinking about machine learning computational prediction models. One is at the parameter level model. So these are the kinds of things that we tend to do experimentally in high throughput. So is it active against this kinase? Is it not active against this anti-target? Does it hit hERG channel? Those kinds of things. And then the more sophisticated model that we're working towards are human level models, where you take these various prediction level models and you move them, you integrate them into a model for liver injury that includes blood flow and the immune system and food and all these other things. So the goal is to really make these human level models that include pharmacokinetics as well as all the individual things that can happen in a body. And then this will give us the ability to not only design based on a set of criteria, but to design based on a therapeutic index. So at what point do we see safety signals versus at what point we see activity on target activities. The tools that have been built in the computational realm to do this at the parameter level model is this data management system, access environment. This is actually a lot of work to put together how to get data from public sources and private sources all into a format that the machine can use. And the pipeline that's been developed, this generative molecular design platform, is called AMPLE. The AMPLE pipeline for training optimization for property prediction models. So we're really working at this parameter level model right now. And just to give you an idea for the scale of things, I'm not going to go into this in gory detail, but just to see here, this is the level of data that we're talking about. 300 gigabytes of numerical data, each data point takes up a very small amount of space. So this is a lot of data, and then the models that get built from all of that data, there are thousands, hundreds and thousands of models. So different ways of slicing and dicing the data to be able to make predictions. So one example, this was just really an early pilot study from of the molecular design platform, where we are taking a target that we have a lot of data for. And we started with aurora kinase A and aurora kinase B. And so the idea of this pilot was to say, can we run this molecular design loop and then the experimental loop? Just does the whole system work in a loop? And so we started with a compound that had been in lead optimization. So there's a lot of data that's built up on it. I think everybody here is probably familiar with it. It's a cancer relevant target. There have been many clinical trials for aurora kinase A selective, aurora kinase B selective, and AB dual inhibitors. So these are all mitotic kinases because they have very different phenotypes. And people have tried to use both in cancer models. But you see that they're very similar to each other. When you do an overlay, many kinases are very structurally similar to each other. So it's an interesting design problem, medicinal chemistry problem, to get selectivity. Another key reason to use it is because we had a big set of data from GSK, and they had potency data on 24,000 compounds for one and or the other target. So the initial libraries, the way we did this didn't use all 24,000 compounds, but we started with the first 3,000 compounds that were made in the medicinal chemistry optimization. So if we started 6 months into this design program, where could we get? Could we get to the same place, or could we get to a better place than years of medicinal chemistry? So that went into the working compound library. And then we set as our design criteria 8 different activities, so parameter level activities. We were looking for the activity of aurora kinase B and A, looking for 1,000-fold selectivity, a couple of common safety parameters, some common PK parameters, and then developability. So this idea that it's synthetically accessible, and then this estimate of drug likeness. So every compound went in through this design criteria, then went through a molecular optimizer, back into prediction, and so forth. And then several compounds came out of that, and then they were made and tested. So this is where this system started. The project started with the red, the first 6 months of data. So these 3,000 data points were the ones that went into the computational model. All 24,000 data points are shown in blue. So you see that most of them did not have selectivity beyond 100-fold. So here's the activity data for aurora kinase A versus the activity for aurora kinase B. And the dotted lines are within 100-fold selectivity. After running through the molecular design paces, the top 250 compounds are shown here in green. So top 250 compounds all met the design criteria for selectivity for B over A. How did we do on the other predictions? The hERG channel was not hard to achieve, and that was actually set at a very a very high bar. So that had to be 100 micromolar or weaker. BSEP, this is a transporter in the liver. That was a little harder to get to, but again, it was set at 100 micromolar, so log a PIC 50 of 4. So achieved close to that. Clearance predictions, solubility predictions, synthetic accessibility score, all in the green range. Okay, so once made and tested, what happened? Well, there are 2 points to it. One is that it turned out that a number of the compounds that were predicted had actually been tested. So they had never gone into the design loop in any way. They hadn't been a test set or a holdout set. They just happened to be in the database, but they were also predicted through the molecular design optimization. So if you take those compounds and you say, what was measured versus what was predicted for Aurora kinase A or Aurora kinase B, the predictions were pretty good, often within 1 log. That would be the dotted line. When we actually went to make the compounds and retest them, however, the predictability was much lower. So there were 2 problems. One, the synthesis of the predicted compounds. Now you're outsourcing chemistry, very diverse chemical synthesis. Took a long time, took months. And second, while the predictions tracked within the data set, those are the pink dots I just showed you, new experiments didn't match the old experiments. So some of these experimental data points were taken a long time ago. Maybe the assay was slightly different. Now using a different vendor, maybe a slightly different assay, the results are not quite the same. So it's probably not all that surprising to people who collect a lot of data, but is very frustrating for molecular design optimization. So what we learned from this activity was that data quality and consistency really matter, that we need to understand how to take imperfect data sets and still use them to make predictions. And also that chemical synthesis needs to be sped up. How can we speed up chemical synthesis? And those are questions that we're working on. So in that chemistry question, we're working to build the experimental loop. So automated assays, even pretty complicated automated assays, is very mature science. But automated chemical synthesis is really going through an exponential phase of growth. And so we have 2 companies that we're collaborating with that are building automated chemistry pipelines. So we can see if we can't speed up that part of the loop so we don't have to wait months to get a single cycle of the experimental loop. Finally, the part that I'm obsessed with is how you initialize the design loop. So what goes into that working library? Because even if you can go very far away in chemical space, using these design properties and these generative molecular design still very much matters what you start with. That's my bias anyway. And so how do we filter things to get into that working compound library? And you heard about some of them today. Virtual screening, DNA encoded libraries, fragment-based drug discovery, high throughput screening, public data sets. This is an area of current focus. So back to Target 2035, how do we enable development of chemical probes? So we use computational and experimental methodologies that initialize that design loop. We need to get better at transfer learning. So we have data on related targets. There's tons of data on kinases, GPCRs. How can we make use of all that data into molecular design for a new target? And then what are the metrics for success for a chemical probe? We have some pretty well-defined metrics for chemical probes. And the last point is that we want to integrate and collaborate with others in this space. 2 companies in particular, Atomwise and Cyclica, have advertised open platforms for academics. There are a number of other companies working in the AI design space that we might be able to collaborate on Target 2035, and also to engage more academic labs and consortia that are working on these problems. And so with that, I just want to thank the ATOM team, my lab, and our leadership in ATOM, Jim Brazzi at Lawrence Livermore National Labs, and Eric Stahlberg at the NCI. And thank you very much. Take any questions.
Thanks so much, Michelle. And thanks to all 3 of you. So please, if you've got a question, throw it in the Q&A box. I've got a question for Alison up front. This could almost have been the selectivity session rather than the chemical biology session, because everyone, touched on that aspect of optimization of probes. Just generally, I wondered if you could comment, as you were making this new, very selective CK2 probe, what's your thinking about when a probe is good enough? I mean, clearly in your mind, CX4945 wasn't good enough. It wasn't very selective. So you're part of SGC. So how do you make a call on when a probe is adequate and when more work is needed?
Yeah, so you touched on kind of the metrics that we go by are the SGC metrics. So it has to have biochemical potency at a certain threshold. I think 100 nanomolar, has to have no off-target activity against more or greater than 30-fold activity off versus off targets, and then it has to be cell active. So we set a threshold of 1 micromolar for cell activity. So those are the metrics we go for kinase chemical probes. We also always provide a negative control compound because we think that's essential for the biological experiments. So we define the criteria that we go by, and I think they're pretty good. And they are certainly hard to achieve. And but we have had success with kinases and epigenetics for them.
Thanks. So it's interesting you mentioned selectivity because, I think you were talking about selectivity within the kinase family, for example. Is that correct? So, and obviously as we now have the tools to ask questions proteomically, and we saw that a little bit in the first session. It's interesting to reflect on what that means. Jordan, as you're using your approaches that you educated us on today, and that I think accessibility is increasing for, how are these proteomic approaches going to help us understand broader proteomic interactions? And as you develop your molecules, how easy is the chemistry to create degraders? We've kind of talked a lot today about covalent and non-covalent. Degradation hasn't really come up, but that's a whole nother chemical modality now. It almost adds another layer of probe requirement. We could have an inhibitor, an activator, and a degrader. Do you want to share some thoughts with us on that?
Yeah, I mean, I think, maybe address the degrader question first. I think it's still kind of the early days in terms of, people thinking about, turning, I guess, low affinity, let's say, photo affinity fragments or, covalent fragments that are, assayed in a proteome-wide respect into degraders. There's some, promise there, I think, if people are excited about it. But, those are still turning those approaches into highly selective ligands. I think, there's still, there isn't yet, I think, a huge success story. There are a lot of industry groups, right, working, I think, very furiously on that. And we'll probably hear some of those stories in the future. In terms of kind of, the first question, sort of just looking at, selectivity using chemical proteomic methods, I really do think, I think it's easy to say that these tools are there now and you should use them. I think it's still easier said than done though, right? There's still, in terms of enzyme class, sort of selective, chemical proteomic tools, definitely those are somewhat siloed within certain groups. The most, I guess, available tools like that are fluorophosphonate and kinase activity-based probes you can buy from, Pierce or whatever it's, called now. But, it's easy, I think, to kind of say in retrospect, oh, well, they should have checked, this type of probe selectivity, using chemical proteomics. But, there may have been no kind of way to do so. So I think, sharing those tools, making it really kind of second nature for this is something that people do, kind of the way SGC, will look at a broad, spectrum off-target GPCRs and kinase, kind of assays when they're developing a probe. Would be really helpful if that could be kind of extended to sort of chemical proteomic approaches and like a core suite maybe of approaches.
Yeah, thanks. So before I go to Michelle, Alison, I guess, based on an answer like that, do you imagine that SGC as an organization is kind of going to move the bar on the criteria for probes as our chemical, biological, and proteomic tools become more accessible? Because the reality is the activity is there, whether or not we're finding it, right?
Yeah. And as was just mentioned by Jordan, we also do GPCR paneling for all of our probes as well. So I think we're definitely infiltrating a little bit. Like my group is part of the Illuminating the Druggable Genome program, and we're making chemical tools for that program. And we are trying our best to adhere to the SGC criteria for chemical probes for that program as well. So yeah, I think our criteria is a very high bar, but it's a good metric.
Thanks. So Michelle, everything a lot of what you talked about with the Atom Consortium really relies on the quality of data. And there's a question, there's a couple of questions here for you, Michelle. You might want to look at Q&A, but one of them, the first one, sort of saying, well, AI and machine learning work good when the biology data is good, and you touched on that a little bit with your aurora kinase example. So, that if we go back to things like dose response data and biochemical assays, I think data quality can be quite high. What are you and Adam thinking about broader sort of discovery data sets, things like proteomic profiling for the covalent warheads we've learned about in the first part of this talk? What constitutes good quality data, and how far do we have to go with actually making some of that data available, like Jordan touched on?
Yeah, it's a good point, and I was thinking about that too while you were talking, that having more proteome-wide data, like Jordan was saying, having something like DepMap for chemical proteomics, it would just be a gold mine for this kind of activity, because it really, I think computational people tend to think, or the data, I don't want to too broad of strokes, but I've often heard data people saying, well, every piece of data is terrible, but there's so much of it that is useful. But it would be really better for us to have really high quality data too. So I think that the best we can do right now, there are a few interesting points in this question that you're referring to. The best we can do right now is have overlap between different data sets so that we can at least scale them to each other, like we do with RNA data. It's all scaled to something. And so subtle changes in chemistry can cause big changes in biology. And so that's why more data is important. And actually ask about, why does Lawrence Livermore National Lab, why are they working on drug discovery? Or their job is to maintain the nuclear stockpile and safety and bioterrorism, things like this. And when you ask Jim Brazzi, he says, it's because it's the hardest problem. Because drug discovery is the hardest computational problem. So if we could just take pieces of it, that's one reason I'm so excited about doing this for chemical biology, because we're not thinking about some of these longer term things like in vivo to human or models to human translation. That's a very big problem. But just, can we take the high quality data and just collect it computationally faster?
And John Tadera has a question in here following from that. So as you predict molecules that may be useful, do you have sort of a chemical intelligence or a chemistry AI there that helps guide decisions, not just about the most active molecules, but the most synthetically accessible molecules or chemotypes to sort of shorten and cheapen the chemistry timeline at the back end?
Yeah, it's a good question. And there's another piece in there too I want to answer. So the synthetic accessibility score is like a guess. But a couple of years ago, when we started working on this with the consortium, I was really surprised at how much effort was still going into figuring out how to computationally design a synthetic route. There's been a lot of progress in this in the last couple of years, but it's not as mature a field as you might think. Compared to biology, chemistry is such a logical, well-characterized science, and still retrosynthetic analysis computationally is still an area of big research. So keeping an eye on that and trying to capture that. And then the subset of John's question is, instead of just making one molecule, can you make a bunch of close analogs like you might do medicinal chemistry? And then that builds the data set, and that builds your selectivity. And then, and actually that's what the automated chemistry is really good at. You find a good molecule, pick a few analogs, and then make all of them automated. The hard thing about automation is doing multiple different chemotypes, which is the goal of the design loop, is not to just give you analogs that a medicinal chemist could design, but to give you things you might not have thought of.
Great. Well, thank you, Michelle. And that answer brings us to an end. So, Alison, Michelle, and Jordan, thank you for this second session. Angela, Jacob, and Damien, I know you're still watching. Thank you for your presentations and answers in the first part of this session. It concludes our panel today. There is a fourth panel coming up on Monday, the 16th of November. As I said, it's an Asia Pacific and Australia friendly session crafted for those people, that will have some of the speakers that we've heard and some new speakers coming together to talk about Target 2035. Thank you everyone for joining us today. I wish you all an enjoyable remainder of your Thursday or Friday, depending on where you are. Thank you.
Thanks, everybody.
Thank you.